right, so protein synthesis is the idea that DNA goes through a process called transcription, at the end of which it makes a molecule of pre-mRNA or primary mRNA transcript. That pre or primary mRNA transcript goes through a section called processing, both of which happen in the nucleus, where it edits the mRNA and adds some features to it. And then that mRNA goes out to the cytoplasm, where ribosomes will read it and use the code found in the mRNA to make a protein. And that last part is called translation. So those are the three big parts of protein synthesis: transcription, processing, translation. Starting with transcription, we start with our molecule of DNA. We're going to end with a pre-mRNA or a primary mRNA transcript. In the nucleus, an enzyme called RNA polymerase is going to unzip the DNA at a region called the promoter. That region called the promoter also has a series of bases called the Tat-top box. It's thymine, adenine, thymine, adenine, thymine, adenine. That's just where it happens and where this transcription is going to start. The DNA then unwinds, and RNA polymerase attaches free-floating RNA nucleotides to the DNA in a 5' to 3' direction. RNA nucleotides are going to be complementary to the DNA, except if you have an adenine on the DNA, you're going to have a uracil on the RNA instead of a thymine. RNA polymerase will continue along adding free-floating RNA nucleotides until it reaches a terminator sequence, and at which point the RNA or pre-mRNA transcript will be released and the DNA will then zip back up. That transcript, that terminator sequence, is then also known as the stop codon. Now, that pre-mRNA that's then made has to go through further processing. A couple things have to happen. During processing, a 5' cap, which is a guanine triphosphate cap, gets added onto the one end, an untranscribed region, and then a poly A tail, a series of adenine nucleotides, gets added to the other end, the 3' end. In the meantime, we have some proteins called SNRPs, small nuclear ribonuclear proteins. Some other enzymes combine together to form a molecule called a spliceosome. That spliceosome will then attach to the pre-mRNA and cut out certain what we call non-coding segments. Those segments that are cut out and removed are called introns. The spliceosome then takes the segments that are left and reattaches them, and the segments that are going to be attached and expressed are then called exons. Once we're done with that process, then we have really what we call our functional mRNA. And the reason for the 5' cap and the poly A tail is to help protect the mRNA as it leads out from the nucleus into the cytoplasm for translation, and to help where the ribosomes are going to bind. Step 3 of this process is translation. Translation is where we're going to start with our mRNA. It involves the ribosomes, and we're going to end with a polypeptide or a protein. So the process of translation involves our piece of mRNA and a small subunit of the ribosome binding to the start codon. The codon is 3-letter base sequence found in the mRNA, and our start codon is always AUG. The start codon also codes for a specific amino acid called methionine. So once the small subunit binds, a transfer RNA or tRNA, which is carrying methionine, will come and bind to the start codon. And the only way that that specific transfer RNA can bind to the start codon is because on the bottom of that transfer RNA is a complementary set of bases called the anticodon, and it will bind or match with the codon. Once that's bound, the large subunit will come and sort of slide its way in and form the whole package of the ribosome. And that first transfer RNA will be stuck in a groove, and the groove that they happen to call is the P site. That's initiation of translation. The next step after initiation is elongation. So the ribosome is going to move to the next 3-letter bases, the next codon. A new tRNA carrying the specified amino acid is going to come and bind. The first amino acid will then be joined with the second amino acid by a peptide bond. Those 2 amino acids will translocate or move over to the new tRNA. The original tRNA will then move to the E site and exit. The ribosome will continue this process. It'll shift down, it reads the codon, the next tRNA comes in, with carrying its amino acid. The amino acid chain gets transferred and connected through a peptide bond, and then the original tRNA leaves to go pick up more amino acids. This process continues until we have the termination step at which the ribosome reaches a stop codon. There are 3 stop codons, and those codons then do not code for any amino acid. They then code and don't code, but a transcription or release factor comes in and it binds, and it causes the amino acid or polypeptide chain to be broken off. It causes the ribosomal subunits to be sort of released and separated back off. And then that protein now, which represents the primary structure, can go on. If the ribosomes are bound to the endoplasmic reticulum, it may end up being transported and exported out of the cell like insulin. If it is free in the cytosol, it may just end up staying there, but it can go through the stages, we know the secondary, tertiary, quaternary folding patterns. Okay?